Hi, everyone. Welcome. I'm pleased to welcome today's speaker, Emily Dolson from Michigan State University, where she's an assistant professor in the Ecology and Evolutionary Biology program in the Department of Computer Science and Engineering. Today, she'll be sharing some of her work studying eco-evolutionary dynamics. As I'm sure you will all become familiar with soon, Emily's work is quite wide-ranging across theory in ecology and evolutionary biology, computational agent-based modeling experiments, oncology, phylogenetic analysis, evolutionary epidemiology, evolution-inspired approaches to problem-solving and optimization in computer science, and most recently, even networks for electric scooter companies. Emily has quickly become a key nexus for collaboration and innovation within our research community and received many recognitions for her contributions. The most recent one I happen to have been there to know about being this year's Outstanding Early Career Investigator Award from the International Society for Artificial Life. I know she has a lot to share with us today, so I'll go ahead and let her get started. Thank you.
Thank you, Matthew. Okay, so I think that was a good introduction to what I'm going to talk about. Hi, everyone. I'm really excited to be here. Thank you for having me. Feel free, if you have questions at any point, to raise your hand. Obviously, there'll be time at the end, but also because it's a wide-ranging talk, if there's anything that you don't have the context for, I want to explain it when you have the question. I'm going to be talking today about kind of two big stories from my research right now. This is actually the first time I've talked about either of these, so you're getting them hot off the press. Both kind of concern identifying ecological interactions for the purposes of understanding their evolutionary implications, and also kind of actually trying to predict the evolutionary implications of ecological interactions. So I'll jump right in. Is that not working? There we go. Okay, so the long-term goal of my research is I want to be able to steer evolving ecological communities to target states. And I think that goal makes more sense if I talk about specific examples. So one really tangible example that I've worked on during my postdoc and have continued to work on since to some extent is cancer. So most of you are probably familiar with the problem of bacteria developing resistance or evolving resistance to antibiotics. Well, we have the same problem in cancer. Often a great new cancer drug will be developed, but it won't cure cancer because the cancer is able to evolve resistance to it. I mean, the whole problem with cancer in the first place is that it's evolving. And so it is evolving on timescales that are very relevant to the treatment. What some of you might not realize is cancer is actually also an ecological community. Most tumors are pretty heterogeneous. They have different cell types in them that do different things. And so if we want to be able to kind of steer the trajectory of this evolving system, in this case to a state where it's dead, ideally, or just where we can control it, we need to factor in the ecology as well. So this is a fairly nice and tractable eco-evolutionary problem with obviously really important consequences for being able to solve it. So this is like a nice entry point. But this problem generalizes. It shows up in other places too. And I'm partially a computer scientist, so I like it when problems generalize. So one slightly more challenging example of this problem occurs in the gut microbiome. In our guts, we have lots of different species of bacteria and fungi and viruses that are all interacting with each other. They're also all evolving on a timescale that is relevant to our well-being. And there's increasing evidence that there are some states that we would like this community to be in more than others for our health. We don't totally know at this point, I would say, what state exactly we want it to be in. But my hope is that by the time the gut microbiome researchers figure out what state we want our gut microbiome to be in, we on the eco-evolutionary theory side can have figured out how to get it. Another example that I study that I don't talk about much today, but if you have questions, ask me. This is like the other half of my research, basically, that I will be not including in this talk. Evolutionary computation is a machine learning algorithm that works basically based on the same principles that make evolution in nature work. And so here we're evolving solutions to problems. Our target is we would like to solve a given machine learning problem. Feel free to ask me later why it's an ecological community. Turns out it is. And so again, it's the same problem. And then lastly, kind of the big picture thing that like actually keeps me up at night and makes me want to solve this problem is like larger scale terrestrial ecosystems, like agricultural ecosystems. As the climate changes, these are going to get destabilized. The bacteria that many of these plants depend on to grow well, and the pollinators and so on, they all need to work together. They all have different ecological interactions. We need kind of collectively the constituent parts of these to evolve in they're able to keep growing and feeding us. And so it's again the same problem, but it's much harder because these things have longer generation times. They're harder to deal with. So I'm not working on this yet, but I care about it a lot. Okay, so that's my goal. You might notice there are kind of two parts here. This first part is about steering evolution. So what is steering evolution? Well, it's basically controlling the course of evolution. And there's been a lot of really good progress on this, especially if we don't factor in the ecology side. So let's think for a second about what this looks like without the ecology. So the most classic example that a lot of you are probably familiar with is the idea of steering the evolution of antibiotic resistance. Again, maybe to a point where the bacteria are dead, or at least where we can control them. So if we have, say, this purple population of bacteria, and we give it this orange drug, we're going to get some new population of bacteria. Likely, those bacteria might be resistant to the orange drug. We don't know for sure. And if we had given them a different drug, say this blue drug, we would, on average, expect to get a different population of bacteria, with a different equilibrium distribution of genotypes. And so the thing that is cool and potentially allows us to steer evolution is this effect, where sometimes the effects of having evolved under the blue drug, and these don't have to be drugs, these could be other treatments as well, will have caused, as a side effect, this population to become sensitive to this other orange drug. This is a concept called collateral sensitivity. It doesn't always happen. It's not guaranteed. But in cases where it happens, we can take advantage of it to apply the different pressures in a sequence that will push the population around the fitness landscape so that it ends up where we're at. To actually do that, there's like a few things that kind of need to come together. First of all, we need to actually know what the fitness landscapes that we're applying look like. So here's an example from a paper I worked on during my postdoc that just finally came out. So these are actually 3 different landscapes created by the same drug. I think this is cefotaxime at different doses. And here, these fitness landscapes are composed of 4 different resistance alleles that the bacteria may or may not have. These specifically are E. coli. And the more yellow colors have higher fitness. And so you can see each of these is a different landscape. If we start out with the bacteria, with the population at a given point in this landscape, it's going to diffuse through it in different ways depending on what order we give them these drugs in. So to sequence those correctly, we also know kind of how long to apply each drug for. How long will it take the population to reach equilibrium so that we know where it is and we can shift to the next step. I also did some work on that during my postdoc. We came up with this technique for basically making the population reach equilibrium arbitrarily fast, assuming we have the right control levers. We borrowed some stuff from quantum physics to make it happen. So this is a tricky problem, but it is addressable. So basically, the steering evolution part of this is by no means a solved problem. There's a ton of super important work to do there, but there's a path to how we do that. The part where I feel like there's less of a path, and that's why I'm excited about trying to solve this problem, is the ecological part. So what would we need to add to be able to apply our steering to an ecological community? Well, we would need to know kind of how the species are interacting with each other, right? And we could think about that as one of these species interaction networks. We could also think about it in terms of game theory or other frameworks for describing interactions. Either of those would work. The big problem is we also need to know the evolutionary consequences of those interactions. And this is very challenging because evolution takes a lot of time. The combinatorics of trying to do all the experiments we would need to do to identify the interactions are really bad. And also, it's inherently challenging to measure any of these things in the lab or the field because our observations are always going to be imperfect, there will be error, and there will be certain experiments we just physically can't do. We can't, for example, later I'm going to talk about an experiment on the effect of partner choice. In some systems, maybe you could knock out partner choice from your mutualism, but things like that are often very challenging to do in lab and field experiments. And so for all these reasons, I think that we need to attack this problem kind of using a plurality of approaches. So a lot of you are probably familiar with the trade-off that you make when you shift from field research to lab research. You become able to do experiments that you otherwise be able to do, and you also do those experiments faster, which is nice. But the cost is you do miss some dynamics that would only happen in the field. There's plenty of fascinating examples of phenomena that show up in the field and the lab because there's extra factors that need to be present for them to happen. We can make the same trade-off by going to artificial life or digital evolution, which are these kind of computer programs that are designed to be a model system for studying biology, Drosophila or Arabidopsis. And obviously, like any model system, experiments we do in these systems might have quirks that make them different from biology, so we shouldn't just assume that things will work the same way. But if we want to get answers to general evolutionary questions, they can be a really useful tool. And so again, we lose some of the natural dynamics, we gain the ability to do more experiments with more controls faster and get the data that we want. And we can make the same trade-off by going to even simpler systems like simulations or pure mathematical models. So I think we need all of these. My personal focus is in the simulations and artificial life / digital evolution portion of this, because that's what and I love collaborating with people who are from other parts of this continuum. So the research in my lab is kind of structured like this. There is this big picture theory goal that we want to address. But that theory goal is kind of nebulous, right? It's hard to think about where we attack that problem from. So the approach that I've settled on is we identify instances of this problem, like I gave you examples of at the beginning, and we try and make progress on those specific instances. So for example, we try and make progress on controlling the evolution of cancer. In the process of doing that, we learn general eco-evolutionary theory that we can then apply to these other problems that we're also working on. It does make for a lot of very different sounding projects going on in my lab, but we're constantly finding connections between them and using things that we learned in one to help inform the others. So the two that I'm going to be talking about today come from the endosymbiosis portion of our research and the evolutionary medicine portion of our research, specifically the cancer portion. Okay, so the first project I want to talk about is this project where we're trying to identify game theoretic interactions between cancer cells using spatial patterns. This is work that's been led by my PhD student, Sydney Leiter, and we've been doing this in collaboration with Maxie Strobel and Jake Scott at Cleveland Clinic. Okay, so a little context on why we're asking this question. So the classic way that people treat cancer is they have a drug that will treat cancer, and they give the patient as much of that drug as the patient can tolerate without having terrible side effects, or without the side effects being too terrible. Often the side effects are still pretty bad. And they keep doing that until, sometimes that works. Sometimes that cures the cancer, and that is fantastic. But the problem is that the other thing that can happen is if our initial population, or a population early on, contains some cells that are resistant to the drug, then we only kill the sensitive cells. We're left with a resistant cell. That resistant cell replicates, and then we're left with a population that the drug does not work on. A classic evolution of resistance problem. What would make sense from an evolutionary theory perspective is to do something more like this. And I should say, this specifically makes sense in the context of costly resistance. So if we're assuming there's a cost to resistance, then the sensitive cells are going to outcompete the resistant cells in the absence of the drug, and the resistant cells will outcompete the sensitive cells in the presence of the drug. So what it makes sense to do is be kind of careful with how much of the drug we give. We give enough to kind of knock down the population of sensitive cells so that it's not too big, because we don't want any of these cells here, really. But we make sure not to actually kill them off so that they're still around to keep the resistant cells under control. So we give some drug, we take a break, we let the population grow back up, we give some more drug, et cetera. We keep oscillating this pattern. And the idea is doing this allows us to maintain the population at kind of a low level where it's more of a chronic problem. It's not going to kill the patient. And preliminarily, this is actually looking really promising in these conditions where there's costly resistance, and we have the right eco-evolutionary dynamics for this. This is a figure from the first clinical trial on adaptive therapy. So this top chart is the people who were on the adaptive therapy protocol. This bottom chart is the people who were not. These X's indicate that their cancer progressed. And you can see the people on the adaptive therapy protocol made it a lot longer before their cancer progressed. And some of them, by the end of the trial, still hadn't had their cancer progress and were still being maintained on this adaptive therapy routine. And I didn't check before this talk, but last I checked, they were still alive. So this is really encouraging. There's a problem. So that works in this case where we have costly resistance. How can we use evolutionary theory to inform cancer treatment when there are other ecological interactions happening? And kind of a corollary, a thing we have to figure out before we can even address that problem is how we can even identify what those interactions are. So one approach that has been developed by Jake's lab is the game assay. So in the game assay, what happens is a whole bunch of petri dishes are plated with different ratios of sensitive cells to resistant cells at different drug concentrations. We grow those up. Well, they grow those up. I have never physically done this, although I've watched other people do it. And they look at the growth rate observed in each of these conditions. They can then plot the growth rate versus the initial ratio of sensitive to resistant cells. And so what that tells us is how the growth rate changes based on the population composition, which will tell us about the underlying interaction. And so they can eventually translate that into game theoretic interaction matrices. So that's great, that works. That is a lot of work though. That's not something that is necessarily realistic to do like for each patient that walks through the door and we want to know how their cancer cells are interacting with each other. So it'd be really great if we could find an easier way. Ideally, a much less time-consuming one. So the idea we had is, can we identify the game theoretic interactions that are happening between these cells using an image from a single time point? So just brief tangent in case anyone is not comfortable with what game theoretic interactions are. So the classic game theory game is Prisoner's Dilemma. In this game, there are two prisoners that are caught. They each have the option to stay silent, i.e. cooperate, or betray, also called defect, their partner. And so if they both stay silent, they both get 1 year in prison. If one betrays the other, the one that got betrayed gets 20 years. The one that did the betraying gets 0 years. It's symmetric. And if they both betray each other, they get 10 years each. This is an example of a two-player game. We can generalize this, though, to include other games that have different dynamics. So instead of thinking about these strategies as staying silent or defecting, we can instead think of these strategies as sensitive and resistant. Because this matrix is symmetric, we can also drop the payoff for the second player. We can just have these represent the payoff for the row player. And we can also further abstract this and just have a variable that represents the payoff for each of these scenarios. So this is sensitive meaning sensitive, sensitive meaning resistant, resistant meaning sensitive, and resistant meaning resistant. Okay, and so as a result of having done these transformations, we can basically plot this space of possible two-player games. So on the y-axis here, we have the extent to which the resistant has an advantage. On the x-axis, we have the extent to which the sensitive player has an advantage. And we get that by subtracting that b term from that d term for the sensitive advantage, the c term from the a term from the c term for the resistant advantage. And so we could further subdivide the space. You'll sometimes see this divided a lot more fine-grained to get different regimes like prisoner's dilemma and stag hunt and stuff. For our purposes, we're not going to worry about that, though. Basically, the way it works out is in this upper right quadrant, we get coexistence. So we have a scenario where each species can invade when rare. Sorry, not species. Each type of cell can invade when rare. And so we should get stable coexistence in that case. Over here, we have a case where the sensitive just does better than the resistant, so we expect the sensitive to win. Here we have bistability. So this is a case where either the sensitive could win or the resistant could win. There's nothing stabilizing it. So in the long term, we don't expect coexistence, but we do expect there could in practice be coexistence for a long time before one randomly outcompetes the other. And then lastly, we have the case where the resistant just has higher fitness always, and so the resistant will win. Okay, so given that, we could ask, do these different interactions produce different spatial patterns? So here are two examples of actual microscope images of cancer cells from these different regimes. I believe here the green are sensitive and the red are resistant. If you see some different spatial patterns, it's not easy. The coexistence are kind of more clumped up and interspersed, which you would expect from coexistence because they want to be near different cells from each other. But there's a lot going on here. It's hard to know for sure. So we wanted to take a step back and start with a simpler system, see what we would expect from first principles before we invest a lot of effort in trying to make this work in the messy realities of the wet lab. So the way we set up this model is we have two types of cells, drug sensitive and drug resistant. They're allowed to reproduce into their local neighborhood. The probability of their reproduction is determined by this payoff matrix. So we have a variable that gives the probability for sensitive to reproduce when interacting with sensitive, probability for sensitive to reproduce when interacting with resistant, probability for resistant to reproduce when interacting with sensitive, and resistant with resistant. And so then to get the probability of reproduction happening, we just look at the local neighborhood of a cell, sum up the number of cells of different types, and multiply by those probabilities. So here we've got one sensitive neighbor, so we add sensitive rate, and then we have two resistant neighbors, so we do 2 times b, because that's the effect of resistant on sensitive, and then we divide by 3 to get the average. Okay, and so if we run our agent-based model with those rules, and we run it with different values of A, B, C, and D that are going to give us different games, we do see kind of qualitatively different spatial patterns. This is arranged in the same way as that game space plot, so this is where we would expect to see coexistence, and sure enough, we see that they're very interspersed with each other. Sorry, this is coexistence. This is sensitive wins. You can see there is indeed more green here. This is resistant wins. You can see there is more pink there. By stability, you can see we're starting to get larger and larger patches where there is a single type present. And so eventually, stochastically, one of those would spread to fill the whole screen, but it would take a long time. Okay, so that's encouraging. Qualitatively, this seems like it might work. Does it work if we actually quantify it? Turns out it does. That is also encouraging. So here, we calculated a whole bunch of different spatial statistics to quantify these patterns. This is one very simple one, which is just within a local area, what is the proportion of sensitive cells? I'm using it as an example because it's very intuitive. And what you can see is we get very different distributions of this statistic in the different game conditions. Bistability and coexistence look kind of similar, but resistant winds and sensitive winds look very different, as you would expect, because proportion sensitive should probably be higher when sensitive is going to win. We did this with a whole bunch of different spatial statistics. I'm not going to get into the details of what all of these are. Just know that they are all different spatial statistics. And basically what we found is that for each pair of games, there are some spatial statistics that can differentiate them pretty well. They're different spatial statistics for different pairs. And so this isn't like a nice, simple rule that we can look at, but there's clearly a lot of information about the games that is provided by these spatial statistics. So we did some information theoretic analysis that I'm not going to talk about here, but feel free to ask me questions if you have them. Kind of just to give ourselves an overall sense of how much information these statistics were giving us. We tried using them to train a very simple machine learning model, just to see if that was enough information for it to do a good job of distinguishing between these regimes. And it did pretty okay. So these numbers could be higher, but they are decent. And I'll point out that actually the main source of error is that it confuses bistability with sensitive winds and resistant winds, which makes sense, because sometimes bistability is going to start to look like resistant winds, and sometimes it's going to start to look like sensitive winds. So overall, our conclusion is that this is pretty encouraging for being able to look at these spatial statistics as a way to differentiate games. The other thing that this plot doesn't capture, but that we've looked at, is the other place that it tends to make mistakes is where the actual point on that two-dimensional space is close to the boundary between games. And so, it's reasonable to make a mistake because it's barely one game over the other. So our conclusion from this theoretical part of this project is that based on this first principles model, it seems like it's possible to identify game theoretic interactions via spatial patterns in a single snapshot. Now, of course, things get more complicated when we try and take this into the wet lab. So that is our next steps. We've got some experimental point patterns from when they've run these game assays. And there's definitely some challenges here that we're working on now. The biggest one is I think our ABM is lacking some dynamics that are happening in the wet lab. And so the patterns just look very different. So what we're doing right now is we're trying to fit the ABM to the experimental data so that we can get enough ABM data to figure out what these distributions should look like so that we can then use them to classify the experimental data. So hopefully that will work. Feel free to look in on it in, 6 months to a year. Okay, that's everything I wanted to say about that project. The other project I want to talk about comes from the endosymbiosis portion of the research. And this is about the effect of partner choice on the evolution of host-endosymbiont mutualism. And this is work led by my PhD student, Kai Johnson. And also all of these collaborators are heavily involved, especially Anya, who created the platform that we're using to conduct these experiments. Okay, so most of you, I'm sure, are familiar with the idea of endosymbiosis, but just in case anyone isn't, endosymbiosis is a situation where one species lives inside another. Those species can interact in a way where they help each other, which would be called mutualism. For example, this cow has bacteria in its gut that help it digest its food. They could also interact in an antagonistic way, which would be parasitism. For example, this cow could get various parasites that would reduce its health, and they would be, feeding off its blood or all sorts of things that parasites can do. So endosymbiosis is really important. It's been kind of instrumental in the development of complexity and life as we know it. And if we want to be able to steer the evolution of ecological communities, steering the evolution of endosymbiotic ones is going to be important. And in particular, one question that we were really interested in is, what is the effect of being able to choose your partner versus not? And we were interested in this partially because we've been doing some research in this system before we added partner choice, and we were curious how much that was affecting our results. But the other reason we were really curious is that this is one of those things that's hard to study in nature. In the field, most systems kind of either have partner choice or they don't. So it's hard to ask questions about what partner choice does to the odds of evolving mutualism, for example. Right, so yes, we have mutualism, we have antagonism. I guess I already introduced these concepts. So what can happen in general, kind of one of the big questions that we tend to ask in this research is how do we maintain mutualism? Because without a mechanism for maintaining it, something that is likely to happen is this. We start out with a population where everyone's mutualistic, they're all helping each other, it's great. But if we get a mutation that makes one of the symbionts basically enrich itself at cost to its host, that is likely to be advantageous to that symbiont at that point in time. It is thus likely to spread. And so, this is an example of cheating, basically. In any system where there is some form of cooperation, there's a risk of it breaking down and cheating arising. And it is a common question to ask, how do we stop that from happening? Or how is that stopped from happening? So in endosymbiosis, there are kind of two big stabilization mechanisms that people talk about. The first is vertical transmission. And so this is when the symbiont's offspring ends up in the host's offspring. So their evolutionary fates get entangled. It promotes mutualism because they both have an interest in them both doing well. That is in contrast to horizontal transmission, which will happen when the symbiont offspring gets transmitted into a different host. The other big stabilization mechanism that people talk about is partner choice. So the ability to choose who you're going to interact with. And our main hypothesis here, based on kind of the partner choice literature in general, is that if the hosts have the ability to choose their partner, it would make sense that they would evolve to exclude parasites and accept mutualists. So theoretically, we would think this would help stabilize mutualism because it gives the hosts basically a way for there to be repercussions against trying to be a parasite. So yeah, we want to ask, how does partner choice affect the maintenance and evolution of mutualism? To do this, we used a system called symbulation, because it's a symbiosis simulation, developed by Anja Vostenaar at Carleton College. The way symbulation works is that there are two types of individuals. There are hosts and there are symbionts. The hosts collect resources every update in order to reproduce asynchronously. Hosts and horizontally transmitted symbionts need resources to reproduce. Vertically transmitted symbionts get a free ride, basically, when their host reproduces. Every individual has a number that is kind of representing its behavior. That number ranges from -1 to 1. And basically, if it is less than 0, it is antagonistic behavior. If it is greater than 0, it is friendly behavior. So for example, if both these numbers are less than 0, the symbiont is going to try and steal resources from the host, and the host is going to try to defend against that. If the symbiont is greater than 0 and the host is less than 0, the symbiont is going to donate resources to the host. The host is still going to try and defend against the symbiont. That's basically a waste of resources for the host. If the symbiont is less than 0 and the host is greater than 0, the symbiont is going to steal resources from the host, and the host is very generously also going to donate resources to the symbiont. And if they're both greater than 0, then the symbiont donates resources to the host donates resources back. The symbiont multiplies its resources by a synergy factor, so they can both benefit. So this is the happy cooperation situation. Okay, so to add partner choice to this system, we added this system of tags. So every individual has one of these. These tags are a sequence of 0s and 1s. And the way we use these to implement partner choice is we look at how well the symbiont's tag matches the host's tag. So here we would say this has a mismatch of 3 out of 5, or 0.6, because it has 3 sites where the symbiont's tag is different from the host's tag. And then there's a parameter we can set called tag permissiveness, which is basically the mean amount of mismatch that hosts will allow from an infecting symbiont. Infection is probabilistic, so all of this is averages. But yeah, in general, that controls how different the tags can be and the symbiont still be able to infect the host. Okay, so what did we see? So our first experiment we did in a condition where we start the population with a mix of mutualists and parasites, and we don't generally expect to see de novo evolution of mutualism, just based on these parameter settings and our past research. We varied vertical transmission rate to vary kind of the extent of mutualism maintenance that we would expect. And then we also varied tag permissiveness. And this first row is a no tag control. And we have the symbiont's behavior on the left here and the host's behavior on the right. We're going to plot basically the proportion of each replicate that displayed behavior ranging from mutualistic in the red to parasitic in the kind of cyan. Okay, so at high vertical transmission rates, we consistently just see everyone be mutualistic because, again, their evolutionary fates are intertwined. This makes sense. As we start to get into the lower transmission rates, we see something a little different. So in the no tag control, these two conditions end up with a mix of mutualists and parasites. Basically, there are some mutualists hanging around. That makes the hosts act friendly. You can see the hosts are actually all being mutualistic. And some parasites are allowed to be there as free riders, basically, and they're benefiting, but they're not swamping out the population. If we have a low enough tag permissiveness, however, the hosts are indeed able to use their tags to exclude the parasites. And so we get more conditions of pure exclusive mutualism than we would otherwise have. As we get into even lower vertical transmission rates, again, we see kind of, so here in the no tag version, there is weaker mutualism and there's also parasitism, whereas with lower levels of permissiveness, we see kind of stronger mutualism continue to be maintained even into these lower vertical transmission rates. Oops. And then lastly, if vertical transmission is too low, no amount of tag permissiveness change is able to rescue this. We get pure parasites. Although if tag permissiveness is low enough, we do get a regime where the hosts are able to just completely exclude all symbionts. And so the symbionts have gone extinct here because none of them are able to get into the hosts. Okay, so that's experiment 1. Right, and our conclusion here is that, here we see kind of narrow region of parameter space, but nevertheless a region of parameter space where partner choice does help enable exclusive mutualism when it wouldn't have otherwise been possible. So that's consistent with our hypothesis. It's honestly not as dramatic an effect as I was expecting, but it's there. The second experiment we did is under a condition that we know historically produces de novo evolution of mutualism. So we start these parasitic, and if left to their own devices, they will evolve to be mutualistic. Otherwise, these plots are kind of the same style. Except here, instead of varying vertical transmission rate is locked in at 0.5, and we are varying tag mutation rate and tag permissiveness. Okay, so without a tag, we get this behavior here. We have mostly mutualistic symbionts with a few parasites and exclusively mutualistic hosts. We get the same behavior as that when the tag mutation rate is very, very low, which makes sense because having a very, very low tag mutation rate is kind of like just not having tags at all. As our tag mutation rate goes up a bit, we do see this zone here, again, where the hosts are able to exclude the parasites. And we get exclusive mutualism when we wouldn't have otherwise. So okay, that's also kind of consistent with our initial hypothesis. However, we also see this zone, which we were really not expecting. This is a zone where having partner choice actually prevents mutualism from evolving when it otherwise would have. And basically what's happening here, we think, is, so the tag permissiveness is still fairly high. It's too high for them to actually fully exclude the symbionts like they're doing down here. But we think basically the hosts are able to exclude enough symbionts that it becomes important. Okay, right. So you can see here the hosts are also being antagonistic in this regime where they wouldn't otherwise. So basically, the hosts are kind of trying to exclude the parasites. They're being more antagonistic to them. They're, in general, not putting up with the parasites. But they're not able to fully exclude them because their permissiveness isn't low enough. And so instead what they do is they kind of create a scenario where the symbionts really need resources in order to be able to hold their own against the hosts. And it basically stunts the evolution of mutualism. So it becomes disadvantageous for them to evolve mutualism, and they get trapped in this parasitism regime. Right, yes, this is just breaking down these different regions. I kind of already talked through them. So the key takeaways here are, first of all, yes, again, consistent with our hypothesis, partner choice can enable exclusive mutualism in places where it wouldn't have otherwise happened. However, it can also lock in exclusive parasitism. So it's more of a double-edged sword than we were expecting it to be. So in conclusion, partner choice basically just has more complicated effects on the evolution and maintenance of mutualism than we were initially expecting. And yeah, if you have more questions about kind of the mechanics here, let me know. I have more slides I can pull up, but I want to leave room for questions. So I'm just going to quickly talk about some next steps. So one of the things we're working on right now in this project is, as we were doing these experiments, we were recording co-phylogenies from these evolving populations of hosts and symbionts. So here we have an example. So this is colored by interaction value. Oh, and I forgot my labels. Symbionts are on the left, hosts are on the right. And we have these records of who's interacting with who. You can see we really have this very small clade of parasites that are kind of infecting most of the hosts. The question we really have about these is, are the tags sufficient to enable there to be specificity in which hosts a given clade of symbionts is going to infect? Because if they were really able to use the partner choice to specifically exclude parasites, we would expect that there would be a pattern where the parasites can only infect some of the hosts. And there might be, it's a little hard to tell. It also definitely varies by condition and so on. So yeah, right now we're working on kind of the right statistics to do to these trees to get at that question of, has this partner choice enabled partner fidelity where there's kind of consistency in which lineages are interacting with which other lineages? The other thing we're looking at is the effect of, oh, shoot. Interesting, okay. I think what happened is my laptop was not charging and it ran out of batteries. So what I will say is we are looking at the effect of partner choice landscapes, where basically the landscape of how mutations will take you from one tag, or from one probability of being accepted by your partner to a different probability are different. Because so here we were comparing tags using what's called Hamming distance, just the strict mismatch count. There's other ways we could compare tags though. And what you would have been able to see on those plots is that turns out to matter. So we get overall the same regimes, but we definitely get them in different parts of the parameter space when there's different mechanisms for assessing how well two tags match each other. And so we're also looking more into that. So yeah, with that, I would like to thank my collaborators and students, and ideally there'd be pictures of all of them right there and my funding sources. And yeah, I will give my laptop battery a second to charge, and then I'll try and bring them back up. But in the meantime, I would love to take any questions. Yeah?
Yeah, so it sounds like
The cube is out of battery.
Oh yeah. I can speak loudly.
Yeah, speak out. Yeah.
Okay, so it was lovely talk, and it's lovely to also know what senior I are doing since I know them. It's super cool, and in the of partner choice. So you have this double-edged sword of you can choose your partner, and that can exclude partners that behave like parasites in the first part, and then it's the second part that locks the endosymbionts into the parasitic way because of it. If I understand, hosts are actively choosing to not interact with them, so the only way to survive is to be a very aggressive parasite. So that's the double function.
Yes, we're pretty sure that's what's happening. Yeah, basically, the hosts stop them from following the trajectory they would need to follow to become mutualists.
Because those organisms are like willing to become mutualists, like hosts cannot or are not willing to interact with them.
Right, yeah, because I mean, obviously, the whole problem is none of these agents like know what's going to happen in the future. They're just trying to make the best choice in the current time. So we know as an external observer that if they hadn't been antagonistic, then it would have evolved to a mutualistic state and everyone could have benefited. But they don't know that. Yeah?
Thank you, Emily. That was a great talk. I'm interested to know, so you talk about the novo mutations and mutualism, how they can emerge. And then you talk about this, there's no space that can get trapped in that's going to force them to go into the first system. So you talk about the justifications behind it. So I'm just kind of lost a little bit. Are you suggesting that because the specific conditions that they're in forces them to have to grab resources from the host that kind of blocks them from transitioning to mutualism? So can you talk a little bit about like what that exactly means?
Yes, okay, so this is the part we're not sure about either. This is like definitely a hypothesis that we're still trying to confirm. But we think what's happening is, okay, so the hosts are being antagonistic. That means the hosts are not just donating resources to the symbionts. So the symbionts can only get resources by stealing them from the host, which requires being more aggressive than the host is being. So that's an incentive to become more parasitic than they started out at. The other way that they can reproduce, other than getting resources, is they can vertically transmit. Here, vertical transmission is at 0.5, so they have a 50% chance of reproducing every time their host reproduces. However, 0.5, well, I mean, so yeah, in some experiments we've done 0.5 is enough to sustain mutualism. In others, it hasn't been. It depends kind of on the other parameters. So we're thinking maybe that's not enough, and they're getting pushed to be parasitic. The other possibility is just in this de novo evolution of mutualism condition, the reason they're able to evolve mutualism is like we get pairs that are kind of gradually de-escalating and becoming less antagonistic. And that might just not be happening because the hosts have started to become more antagonistic, and then that further incentivizes the parasites to become more antagonistic, and it just kind of becomes like an arms race in the opposite direction. Yeah.
I'm curious about from the portion of the talk at the beginning, discussing the game theory among the cancer cells and detecting that using an image. How do the cancer cells disperse? Are they, when they're proliferating, do they kind of move around in that space, or are they kind of like really where they are is a very close reflection of where their parent was?
Fantastic question. Okay, yeah, so how do the cells move around? This is actually what we think is the crux of our current problem in matching the agent-based model to the wet lab. So it depends on the type of cancer. Cancer, the offspring cells basically just end up next to the parent cells. In others, though, they'll move around a fair amount. And so, it really just depends on the cells. Yeah?
Sorry if I missed this, but in the simulations, it sounded like you were saying that at the end of the simulation there's just like a fixed sort of state, and it's not like it ever oscillates, right? Like it's not like any state was ever like, oh, it's parasitic for some generations and then useless for some generations.
Oh, fantastic question. Right. So are the states at the end stable? In all the conditions I showed here, they're fairly stable at the end. There's kind of an attractor that they're falling into. That's definitely not always the case, though. We've seen other conditions where they're very much not stable and they're oscillating. And yeah, in fact, some of the other work Kai's doing now, well, oscillating because we're sequentially applying stress. And so, we're killing a bunch of hosts every few updates. So that forces oscillation. But yeah, actually, gosh, do we ever have examples of like organic oscillations? There are some, I think, but it's not as common as you would think. And that might be due to the specifics of this system. Because obviously, there's a lot of situations in nature where we see oscillations. Yeah?
So in the first part, we talked about adaptive therapeutic.
Mm-hmm.
I mean, this is like giving the proper dose, so you maintain a mixture of sensitive against cells, so kind of like maintains the status of the cancer within comprehensive reach. But the thing is, for a high costly sensitivities, if you kind of apply the same pressure again and again, doesn't like come into play the compensatory mutation. So each time you apply the drug, then the sensitive strain might be a bit more fit than the last time. So you maybe are actually promoting the growth of the sensitive strain to match up to you end with a problem you cannot solve in that way.
So like, are you basically slowly evolving resistance among the sensitive cells too? Mm-hmm.
The idea is used to like keep using your antibiotic until like the whole thing has died, and not try to like give time doses because that would promote the sensitive strain to get the resistance and then be out more fit than the one time immunosuppression.
Oh yeah, okay, great question. Right. So why is this different than the antibiotic resistant case, such that it would make sense to use a different strategy? So there's a few reasons. A big one is that people can't share their cancer with other people. So a lot of the issue with antibiotic resistance is that if you give someone a small amount and you add some selective pressure for the antibiotic, then those slightly resistant bacteria might get into another person, and could over time have a population of bacteria in our species that are becoming more resistant. Fortunately, because we're not Tasmanian devils, that's not going to happen in cancer. The other reason is, I had it in my head. What was it? There was totally another reason I was thinking of. Yeah, so I mean, in this case, the other reason it's different is, okay, I think what I was going to say is it's definitely also the case with these collateral sensitivity strategies that sometimes you can't keep it under control forever. There are some where the best you can do is just delay it because you are always like gaining a little bit of resistance that you can't collateral sensitivity or later. And that all comes down to the specifics of the fitness landscape. And so I think this cancer that this clinical trial was in was castrate-resistant prostate cancer. Which happens to be like a really, really good fit for this strategy. Like the sensitive cells are very sensitive. The resistant cells can be easily controlled by the sensitive cells. And so the strategy of maintaining it at a low level works there. There's definitely a whole bunch of other cancers where it's not going to be so simple.
Let's go ahead and thank our speaker.
Thanks, everyone.